Okay, hello everyone, and welcome to the inaugural lecture of Professor Atticus Hainsworth. I'm happy to see so many people here and online.
Sorry, that's
Let's
me.
stop the echo. Yeah, so good evening to everybody here and online and working at home, watching at home, and those on catch-up. So a little bit about Atticus. Professor Atticus Hainsworth, he's a leading international expert on cerebral small vessel disease. So he completed a BA in natural sciences at Cambridge in 1985. He obtained a PhD in physiology and biophysics from the Rush Medical Center in Chicago, and then undertook a postdoctoral fellowship in Oxford, a welcome travelling fellowship in Paris, and a technical post at Leeds University Medical School. And in 1994, he became a lecturer in the new University of Greenwich. In 1998, he moved to the School of Pharmacy in De Montfort University in Leicester, and that also included a sabbatical at Mass Gen Hospital, which is the leading teaching hospital in Harvard. So in 2006, he was recruited as a senior lecturer at St George's and promoted to reader in 2016. So after a very varied and international early career, we're very glad that Atticus decided to stay here. He's been here for the last 19 years. Obviously, his feet got tired, all that walking about from different places. So at St George's, he's worked on pathological processes of disease that underlies cerebral small vessel disease that causes lacunar stroke and is implicated in vascular causes of dementia. This is a disease with very high impact on quality of life and the economy. So it's a very important area for clinical research. So I probably met Atticus probably around 2008 when we shared responsibility for organising and delivering the clinical neuroscience BSc course. I think we did that for over a decade. That was very enjoyable, and contact with the students, and Atticus was very well liked by the students. And over that time, I'd sometimes get a 6-foot shadow at my door that was invariably followed by the words, Atticus's cheery catchphrase, "Dear chap," and so the need to get something done. I think I've progressed to being old chap now. I've been peripherally involved in some of Atticus's research because I coordinate the research using MRI. So he's brought us plastic boxes containing interesting tissue samples to scan in relation to his histopath studies. And more importantly, there was a PASTIF study, a clinical drug trial to look at brain blood flow in patients. And I'd like to point out clinical trials are arduous, hard, so it's a real achievement to complete this successfully. And I'd say the breadth of his scientific expertise, from detailed histopathological studies to transgenic models of disease and clinical trials, is one of the great strengths of Atticus's research. And there are several criteria leading to promotion professor, and Atticus's role external to St George's include lead of the MRC Dementias Platform UK Vascular Experimental Medicine Group, deputy editor-in-chief of the Journal of Cerebral Circulation, Cognition, Behaviour, and advisor of the Alzheimer's Association, to name but a few. So he's a leading international researcher in small vessel disease, demonstrating leadership and impact. And his research and recent successful grant awards make cerebrovascular disease a strong neuroscience research focus in the newly formed City St George's. So I'm looking forward to hearing about Atticus's 40-year journey as a neuroscience researcher. So Atticus, not so old chap, please come up, and everybody, please join me in welcoming Atticus to give his inaugural professorial lecture.
Wow. Well, thanks so much, Professor Howe. And I've practiced this talk, but it's all gone out of my head in the heat of the moment. And what sort of visits itself upon me is what an honor. What an honor to have all of you, these wonderful family members, friends, some dating back quite a few years, and colleagues here this evening, on this gorgeous summer evening, giving up your time. I feel as if I've I've got the MasterChef trophy and the Strictly glitter ball, one in either hand. Evidently lost on most of you. Let's just check. Can they hear me online? Of course, they can't say anything because they're muted. So who knows whether they can? Heather knows. I'm Atticus Hainsworth. Here's where I work, and the handy tube map as to how you would get there. And let's take a look at what's in this box. So that's what 1.4 kilograms looks like, give or take. Bought this at the weekend, and I claim that I can still say the human brain is the most efficient supercomputer that's ever existed in terms of its speed, its plasticity, and its parallel processing ability. We have supercomputers now. They work pretty well, but they're huge and they take a huge amount of power. And frankly, they're not as good as the one you've got between your ears. And all they take is a bit of blood. All the energy they need is a bit of blood. In fact, they need that much blood every minute. That's what's going through your think tank and mine. And yeah, so it's fairly small. I'm just going to get one in my hand. It's a little bit Tommy Cooper, a little bit for the older folks. So I'm going to advance a little bit. No, that's the wrong thing to do. So you can briefly dwell on my conflict of interest and disclosures. And then I'm going to tell you, when I came here 19 years ago, back in 2006, as a new blood research lecturer, I think I considered myself an expert on the molecules of ischemic stroke. The sort of molecular changes, the drugs that we had to play with back then, and the thing with that supercomputer, that 1.4 kilograms, is it works just fine as long as it keeps getting its blood. If that blood supply is interrupted, even for 15 minutes, the consequences are quite serious. The molecular changes are I quaintly say, a pitched battle. And if it stays that way, if it stays deprived of blood for a few more hours, they won't be reversible. And that's called an ischemic stroke. So I came here, I remember meeting the then professor of anesthesia in the corridor. And he said to me, "In what are you a big man?" And I'm not sure what I said, but what I would have thought was molecular neuropathology of stroke. And indeed, I produced diagrams like this. Don't sort of read it or take it in. The point is it's hugely detailed, and we really believe that. I did that without irony. That was we thought that was what was happening in the molecular pitched battle of ischemic stroke, and that there were various molecular targets that we could direct drugs against, and frequently did, without huge success. So we talked about things like neuroprotection and neuroprotective drugs, which you can't really talk about much now. So anyway, I was a big man in molecular ischemic stroke. But it was not always thus. I had very small beginnings. That's me as a 16-cell embryo. Guy Whitley probably knows the name for that. And most of the credit for that goes to these two. These are Lucy and George Hainsworth. There they are in Rome on a scholarship my dad had. Both practicing artists all their lives. And that's about when they would have been initiating me. And there we are earlier this year in their garden. I think they're watching online. And I had a fantastic childhood in rural Yorkshire, in a village, and I'm going to be talking about scientific mentors. And really, the first is my old mum. There she is with my sister Kate and me. And she had a decent grounding in science, and we did all sorts of stuff together. We dissected a dead badger we'd found on the road, and we'd go out and look at the night sky through my binoculars. She told me the facts of life when I was about 4 and got them more or less right, or as far as I know she did. And I went to the village school. You can see if you can spot me. There I am. Some fantastic teachers, Mr. Bertram on your left. And I went to the local comprehensive in Knaresborough. Again, some inspirational teachers. I'm just going to pick out one, Mr. Wilkins on the front row, who has a look of John Cleese. And his son was in my class, and he's in the audience right now, I'm honoured to say. So I worked pretty hard at school, and I got into Cambridge, and Cambridge was like an awakening for me. It was a whole new start. It was amazing. I was dazzled. I was this young lad from rural Yorkshire, and all these sort of new ways, new thoughts, new opportunities, a myriad of doors that could be gone through, and I went through quite a few of them, and arguably went through perhaps too many of them, because on my final degree certificate there was a reference to a Southern African cleric, which rather shot my chances of doing a PhD. Anyway, in the course of my final year, I'd come under the influence of a rather odd, rather remarkable man called Steve Ladkey, who I think is online listening. And I did my final year project in Steve's lab. And after graduation, I went back and did a bit more. And we did some pretty heavy duty biophysics. And we even got it published. And I saw my name on a paper. And I think by then I was hooked. I had got the research bug, and it never quite left me. So I think that helped me to get a PhD place in Chicago at Rush Medical Center, where I was supervised by another really quite odd and remarkable man called Rick Levis, sadly no longer with us. There he is with his 4th wife, Juanita. I think he died of exhaustion. And Rick was an absolute master of high resolution electronic recording. And we were using what was then quite a new technique called the patch clamp technique. I was a total convert to it. I thought it was wonderful. And you could measure the current through a single protein, a single ion channel protein. And these currents you're looking at, these are electrical current traces against time. Those are literally a billionth of the current that's flowing in your mobile phone device, or through this PC that's running the talk. And we were fascinated by that, and we thought we could analyze the different conductance states of the protein molecule. And we were doing all this in a small muscle, tiny little skeletal muscle in the head of the American lobster, which in fact left the tail more or less intact and quite edible. So I returned from Chicago to the UK, well nourished, and quite an experienced biophysicist. And so I got a first one, then another quite prestigious postdoctoral fellowship, first in the Dreaming Spires of Oxford, and then the Dreaming Spires of Paris, with these two eminent physiologists, Fran and Philippe. And that should have been the sort of launch pad for my career to go to quite good, on a quite good trajectory. I was going to be launched, but that didn't happen. The two projects, my data didn't support the prevailing hypothesis. I don't think I was a very good postdoc. I didn't get on terribly well with my supervisors, and it all sort of fell apart. I essentially packed in science. I decided it wasn't for me. I wasn't that good at it, and I went back to live with my old mum and dad. Did a lot of painting, and I was going to go to art school. I'd got into a London art school. I produced tons of paintings. There is one. This is I think from my Cubist period. And yeah, so I was cranking out these paintings. I still have a few of them, if you're interested. And I traveled around the world a bit. I'd saved money from my Wellcome fellowship, which was very a generous fellowship. And I probably did a bit of thinking as well. And long story short, I got a new lectureship at the then new University of Greenwich. This is back in the early 90s. And there are the gleaming spires of Woolwich High Street, which is where our department was. And I absolutely loved it. And I think I sort of blossomed. I'd been in some pretty high ranking places and felt like an absolute minnow, a plankton. And here I felt like I was quite a needed part of the team. I felt like I was valued, and that I was quite a decent sized fish for this new place that was kind of finding its way in the academic world. And I started to develop my own research a bit, and I got a Wellcome Career Development Award to go and work with Steve Ladkey again. Got some grant money, got some equipment. Clearly, this Ladkey character was an influential person in my career. And around this time, another influential person came into my life. This was the new lecturer in nutrition at the University of Greenwich. That's the first photo that I have of Hilda. There she is in the Yorkshire Dales, looking her usual radiant self. And this is a science lecture, but allow me a little personal interlude. I'm really grateful to have had Hilda in my life, and she's still in it. And I just want to thank her infinitely for all the support and advice and friendship and love that we've shared in the last 28 years. There we are earlier this year, again up in the wilds of Yorkshire. In due course, two other very influential people came along. There they are with my old dad. They're shouting a rude word at the cameraman, which is of course me. And again, at risk of labouring the personal theme, I just feel hugely privileged, grateful, proud. Proud doesn't do justice to what you feel about your kids. To have had my sons Matthew and Fergal in my life. Matthew's in the audience, and Fergal, I think, is online. He says he is anyway. And there they are earlier this year. So thank you guys. Back to me. So by now I was at De Montfort University in Leicester in a school of pharmacy, and they let me go for a sabbatical to Harvard Mass General Hospital to kind of reskill. I went to the lab of Mike Moskowitz, and I was going there to learn what we think of as the molecular biology techniques, some of which hadn't existed when I was a student. So I was trying to relearn, I guess, trying to retrain. And I got reasonably good at protein biochemistry. And I especially got into this immunohistochemistry technique, which is a way of taking a molecular probe to pick out a particular cellular molecule, a biological molecule, and make it visible. Make it so you can see it down the microscope. Here we're labeling two different molecules. We're labeling one red and one green, and we can see the overlap in yellow. And I got quite good at that, and I've been doing it ever since. So I came back to the UK, and I was probably ready to look for how to take my research life forward. And an unlikely fairy godmother came along called Hugh Marcus. Hugh hired me in 2006 as a new blood research lecturer. He took a gamble on me, and I'm very grateful for that. And so there are the dreaming spires of Tooting, where Hugh was a daily visitor. And Hugh and I we didn't have the easiest relationship. We had something of a bipolar relationship. But he said a lot of useful things. And one thing he said was, don't dabble. Do a few things and do them well. And that was a good thing to say, because I was a bit of a dabbler. And so I've focused ever since on cerebral small vessel disease. Pretty much I have anyway. I don't think I've dabbled too much. I've followed Hugh's mantra, and I've stuck with small vessel disease. Now, I want to get this bit right. So it's something of a Cinderella disease. You don't hear that much about it. You hear plenty about stroke and Alzheimer's. This is very common, but I suspect few people on Tooting High Street would know about it. By the age of 65, half of us have got it to some degree. It's the cause of what are called lacuna strokes, which is 25% of clinical stroke. And it's now acknowledged to be the main vascular cause of cognitive impairment and dementia. And that's big. That's like a huge territory of clinical need. And we still don't have targeted therapies. We don't have drugs, but we're doing our best. We're getting a little bit closer. Now, to the microscopist, here's what it is. It's a fibrosis. It's a stiffening of the artery wall. These are two little arteries in a human brain taking blood deep down, and they've got this greeny blue stuff in the wall, and that shouldn't be there. That's fibrotic material that's accumulated in the wall of the artery that is making it stiff and making it so it can't quite dilate. And that's bad, because this is a cross-section through our huge human brain. They wouldn't let me bring in a real one. This is a resin-filled image of one. And in the little red ellipse, I've highlighted some of those deep penetrating arteries. Our huge human brain, we need a lot of them. A mouse doesn't really need them. They're diving down from the surface of the brain to those deep brain areas that need lots of blood. They're thin, they're about the thickness of your eyelash, and they're maybe 5 or 6 centimeters long. So they're a long, thin supply pipe. And for the brain, they're a smart supply pipe. They can dilate, they can adapt how much blood is going through according to the need of the downstream territory. So if I waggle my right hand, the bit of cortex that controls my right hand gets a bit more blood. Or I hope it does. The appropriate little arteries dilate a bit and let a regional increase in blood flow happen. Trouble is, if the arteries are all fibrotic and stiff like these ones, they won't be able to do that. So the downstream bit of brain won't get quite enough blood. This is not a stroke. It's not a lack of blood. It's just an undersupply of blood. And over the years, the brain is quite unforgiving and builds up those chronic lesions over the decades. There's how they look to the radiologist. There's an MRI scan showing those fluffy white lucencies that are the result of this chronic underperfusion. Not enough blood over a 70-year lifetime will result in this accumulated underperfusion damage. And that can't be good. And so just to labor the point, our huge human brain has this slab of white matter in the deep area. There it is with a red arrow. And it's all the connections. It's the nerve fibers connecting one neuron with another, connecting the cerebral cortex with my right hand and toe and everywhere. And they're coated with this stuff called myelin that makes them white. They're the connectors. And we're talking about damage to that region. We're talking about undersupply of blood to that region, leading to damage to the fibers and the myelin. On the right, you see a human-made supercomputer. And I put it to you, if we ripped out those yellow cables that are the connectors, it wouldn't be quite so super. That would be a great way to damage, to impair the functioning of that human-made supercomputer. And in the same way, damage to our white matter is a source of lost brain function and impaired cognition. So we, as a group, we got quite good at this. This has been what my lab has focused on in a large degree. Here are some more examples from a recent review. The red arrows are pointing to the bad bit, the fibrotic material in the artery wall that's making it stiff and undistensible. And so I made quite a career out of this, I suppose, in my time at St. George's. Round the time I'd just been hired, I went to visit Margaret, who I'm delighted to say is in the audience, and she showed me some human brain, and we worked on quite a few aspects of it ever since. And she said something useful. I'd spent the previous 10 or more years studying cells in culture, and I was starting to think about animal models relevant to disease. But Margaret says Margaret, when we were looking at this human brain tissue, she said the answer has to be in there. The answer to what's causing small vessel disease may be in your animal model or your Petri dish, but it has to be in the human brain. And we just have to be smart enough and lucky enough to figure out what it is. And so that's what I've done for most of the time I've been here, with the help of Les Bridges and a talented technician called Joycelyn, who I think was going to try and be here, but I can't see her, so maybe she didn't. And we used our molecular probe technique to visualize different aspects of brain tissue. We could see the nerve fibers. Here they are with the red arrow. They're those little sort of spaghetti strings. We were able to label them. Alternatively, we could label the brain support cells, the astrocytes. There are some examples of them. We could label the brain's immune cells, the so-called microglia, which patrol around the brain tissue, looking for harmful molecules to neutralize them. And we could label the little guys called oligodendrocytes, which are the ones that make the myelin. So this is within white matter. It's actually quite denuded. This is rather sick white matter. Those are the remaining mature oligodendrocytes. We're feeling that if we could beef them up a bit and make them a bit more active, we could maybe repair some of the white matter lesions. And so we've published some of this. We've done a series of published studies, hypothesis driven, where we've gone into human tissue with a question. We've labeled our molecular markers, and we've come out with an answer. And you can see we've made it quite a cottage industry. I'm pleased to say that a lot of these co-authors are students who've done projects in my lab, and some of them are here today. So alongside all this human brain tissue, I did look at some animal models. I do think they have merit. I wrote this review with Hugh that was quite influential, and I looked at quite a bit of rodent brain myself. This is a rat brain, but yes, the penny slowly dropped that this really doesn't look very like the human brains that I've been showing you. It's small. The rat's brain is about the size of my final joint of my little finger, and it really hasn't got very much white matter at all. So for that reason, I've gravitated towards larger species, and they're great. They have bigger brains. They have brains with lots of white matter, and they have blood vessels that really look like human blood vessels. The ones I'm going to concentrate on right now are the pigs, which have sort of taken over my life. We've got this project running with the Roslin Institute, and notice on the left the beautiful pig brain in coronal section. When you go and look at it down the microscope, the blood vessels look very like a human's blood vessels. And so this has been about, as I say, over 10 years really working with the Roslin, and with the help of BHF, they finally funded us to do this. We've made a transgenic pig strain. We've put a human-made gene into the pig's genome, carrying encoding a peptide called endothelin-1, which is one of the most potent vasoconstrictors known. It really constricts little arteries. That was why we thought it was relevant. The Roslin are good at this. They do a lot of it, and you can see these are the genomes from the first litter. All the animals have the transgene except one. Number 5 is a wild type where the injection didn't work. But there's a neuroscientist with the first litter way back in 2022, feeding them some banana. They're really healthy, happy pigs. Because the great thing with this transgene, the Roslin did a blog about them. The great thing with this transgene is you can turn it on. It's not on all the time. They get born, they grow up, they're perfectly healthy. When they get to be adults, you can turn on the transgene by giving them a drug in their diet called doxycycline. When they get the doxycycline, the transgene gets activated, their ambient endothelin levels rise, and they become transgenic pigs. Trouble is, how do you get them to take the drug? Doesn't taste very nice, doxycycline. So that was something of an issue. And so turns out it wasn't so much of an issue after all. So have the folks at home seen that? Awesome, thank you. I could play it again. No. Oh no, there we are. So very much aided by well, a big team of helpers, but particularly these 3 chaps, Simon Lillico at the Roslin and Daniel and Joe based here at St George's. Really with their help we've pushed this forward, and in a nutshell, the pigs have heart small vessel disease. When we look at their hearts, they're enlarged. The transgenic ones have enlarged hearts with fibrotic arteries in the heart wall that we're exploring, and we're moving on to ask, have they got any sort of brain small vessel disease phenotype that would be interesting for our studies. So moving on to the clinical trial that we did. This is like the final chapter. And this comes from a kind of, they call it a light bulb moment. Back in 2014,
And I remember thinking this. It was in a meeting with Margaret Aziri, Les Bridges, and Hugh Marcus, and it was a realization that we could keep doing these human tissue studies, and if we were lucky, we'd discover a molecular target relevant to the cause of small vessel disease, and then we design a drug to hit the target, and we check whether it was tolerated in humans and whether it had bad side effects, and then we'd be ready to put it into patients. But that, frankly, would be about 3 times the remaining career I had left. So I became an overnight convert to the repurposing concept. That's where you take an existing drug that's being used for another disease, and you say, well, based on what I know about that drug, can we use it in my disease? And so after a lot of head scratching, we actually decided that the drug we wanted to repurpose was a so-called PDE5 inhibitor. The best known is sildenafil, made a lot of money as Viagra. And this is a serious area, so I wouldn't want to trivialize it by kind of making any schoolboyish reference to its normal indication for erectile dysfunction and sexual dysfunction. But our idea was it dilates blood vessels in erectile tissue in men and women. Might it dilate blood vessels in old people's brains? Might it be the answer to that lack of blood flow in deep brain areas if we could dilate their little arteries and let a greater flow of blood come through? So we got this funded by Alzheimer's Society and the ADDF. We were funded to do a phase 2 randomized, double-blind, placebo-controlled trial. It was a crossover design. So all the patients got the drug and the placebo, but they were randomized to the order in which they got them. So one visit they get tadalafil, one visit they get placebo, and none of us knew which was which. Notice we're using tadalafil instead of Viagra, as it has a slightly longer half-life. Our power analysis said we needed 54 patients, and we got them, I'm pleased to say. There's a nice picture of a chemical, that's tadalafil. There's a nice picture of Jeremy, who has been my clinical mentor for the past 13 or 14 years, I guess, and was the co-PI on this trial. And so for a good 4 years, probably a bit more, this was a huge amount of work, getting patients recruited, analyzing the data, figuring out what it meant, and writing it up. It was negative. It was resoundingly negative, as I have to tell you, most clinical trials are. It was a huge amount of work. It was a fantastic learning experience. We've gone on, we've taken that knowledge and we'll go forward with it. But it clearly, giving the drug did not increase blood flow over placebo. There we are. There's the Pastis team, or some of them. And we published our negative finding, and it's been, I think it's been quite influential in the field. We've all learned from this. Our original, the Pastis trial, was an acute exposure. It was one dose of drug. Patients came in, they got the drug at lunchtime. We measured their blood flow at 3 in the afternoon. And as you see, the drug didn't do anything much. Now we're taking this forward. We have a large grant submitted with a Korean drug company. This is a collaboration with our colleagues at Imperial, Alistair and Fatima, using a different compound that they claim is more potent and gets into the brain better. So I think that, I personally believe that story has more mileage in it, the use of Viagra-like drugs for possible use in dementia. Okay, I'm nearly done. So in a way, I'm most proud of this of all. I've mentored, I've sort of trained quite a lot of junior colleagues over the years. There's a list. If you're watching and I've forgotten to put you on, I deeply apologize. These are the photos of the ones I could get from LinkedIn. And I'm going to pick out just one, which is Mr. Anand Steyer, who's at the front there. He's currently serving his country in the Palestinian West Bank, where, as far as I can tell, he more or less runs a big hospital as well as doing a full neurosurgical job. But I'm really proud of all of them. I'm proud of that list of names because I've done what I've done. But there's also all those colleagues out there doing clinical careers or biomedical research careers, and I've helped them a bit. I've got them going. I've given them a bit of a start, and so a key message I'm giving myself is pass it on. If you've got it, give it to someone else and let them go forward. Blimey, I'm done. This is my wonderful list of collaborators and funders, and thank you very much.